Hello, in this section we shall be dealing with purine synthesis and catabolism. Purine is a heterocyclic aromatic organic compound that consists of two rings, a six-membered nitrogen and a five-membered imidosyl ring fused together, and it is water-soluble. We have two purine molecules. They are adenine and guanine. Nucleotides can be recycled from nucleic acid and cofactors that are broken down, and can be synthesized in de novo from several amino acids, and no specific dietary requirement is required. The synthesis of purine requires first the formation of the sugar phosphate backbone onto which the bases are built, wherein in pyrimidine we see that the bases are attached, or they are first formed and then they are attached to the sugar bases. For the formation of purine we require glutamate, glycine, tetrahydrofolate and aspartate. To begin with, we, the cycle begins with the pentose phosphate pathway, which is converted into ribose 5-phosphate, which in turn gets converted to 5-phosphoribosyl 1-pyrophosphate, which is in short is known as PRPP, in the presence of the enzyme PRPP synthase, and which in turn is converted to 5-phosphoribosyl 1-amine, wherein the glutamine is converted to glutamate in the presence of the enzyme glutamine PRPP amido transferase, and this is the first committed step. And from here you see a series of steps being followed, that is 10 to 14 reaction occurs to form the final product, that is inosine 5' monophosphate, which we will not be going into detail into these stages. But let me put what are the important groups which are required. You can see here we require the glycine and the ATP, N-time, formyl, THF. Again, we require the glutamine and ATP, carbon dioxide, aspartate, ATP, and N-10, formyl tetrahydrofolate. So, a series of reactions with the utilization of ATP and carbon dioxide and other amino acids, you see that the inosine 5' monophosphate is being produced. So this is the structure of INP. This is the ribose sugar. You can see that the nitrogen is donated by the glutamine, and the nitrogen and carbon is donated by the glycine, and the aspartate supports the amine group, and the carbonyl is provided by the bicarbonate, and the CH group are produced from the formyl tetrahydrofolate, or in short known as THF. So once a product, whatever we mentioned in the previous slide, that is the ribose 5-phosphate inosine monophosphate is produced, it then converts itself to form ANP and GNP, which are the purine molecules. This can happen wherein the ribose 5-phosphate INP is converted to ribose 5-phosphate adenosuccinate in the presence of an amino acid aspartate, wherein The entire aspartate molecule gets attached to the ribose 5-phosphate sugar molecule and utilizes GTP. In the next step, you can see that the ribose 5-phosphate adenylosuccinate is converted to AMP by the removal of the fumarate, but still the amine group attached. This component is AMP, the final component, which is being produced from the IMP. IMP is also involved in the production of GNP. So, in the production of GNP, first the water is oxidized to carbonyl and then again are replaced by the carbonyl with the amino group from the glutamine, and this reaction requires ATP. And finally, you can see that guanosine monophosphate is being produced and the amine group is donated by the glutamine, which in turn gets converted into glutamate. So, in the first step, you all saw how the water is oxidized to the carbonyl group, and then again it is replaced by the carbonyl group with an amino group from the glutamine, and the reaction requires ATP. Thus, the GNP is being reduced. Coming to the purine catabolism, we begin with the ANP, which can be processed to adenosine or it can be processed to INP in the presence of the enzyme ANP deaminase, which is followed by the removal of the amino group. Similarly, the adenosine can get converted or processed to inosine in the presence of the enzyme adenosine deaminase, which is known as ADA. ADA is a very important enzyme. Any deficiency or destruction in the gene of ADA can result in severe combined immunodeficiency disease, which affects 15% of the whole population. So we can see here the adenosine can be processed to inosine, or even the IMP can get processed to inosine with the removal of the amino group. The inosine formed is then converted to hypoxanthine in the presence of the enzyme purine nucleotide phosphorylase, wherein a ribose 1-phosphate is being removed, thus forming hypoxanthine. The formed hypoxanthine can enter into the salvage pathway, or it can get converted into xanthine with the production of hydrogen peroxide in the presence of the enzyme xanthine oxidase. This enzyme xanthine oxidase is also important, wherein you know that the purine can result in the formation of too much accumulation of this purine, can lead to the formation of a disease known as gout. So, drugs have been synthesized against the xanthine oxidases to prevent that particular disease. So, as I said earlier, hypoxanthine can be get converted to xanthine, and further it can get converted into the uric acid with the liberation of hydrogen peroxide. Large quantity of production of uric acid can be harmful. As I said that earlier, it can lead to the production or the formation of a disease known as gout. In the next step, you see that GMP is processed to guanosine, and this guanosine is then processed again to guanine, which can enter into the salvage pathway. Otherwise, the guanosine, which are formed by the enzyme purine nucleotide phosphorylase with the removal of ribose 1-phosphate, can get converted to xanthine in the presence of the enzyme guanosine with the removal of the amino group, and thus forming the xanthine. The salvage pathway means, you can see here, both hypoxanthine and guanine can in turn get converted to IMP and GMP, wherein the PRPP is converted to inorganic phosphate. The IMP in turn can be converted to GMP, or in turn it can be converted to AMP, and the AMP and GMP can lead to the formation of again the synthesis of the purine. With this, we come to the end of the synthesis and catabolism of purine metabolism. Thank you.